Microbial metabolism is the means by which a microbe obtains the energy and nutrients, example carbon, it needs to live and reproduce. Microbes use many different types of metabolic strategies, and species can often be differentiated from each other based on metabolic characteristics. The species metabolic properties of a microbe are the major factors in determining that microbe's ecological niche and often allow for that microbe to be useful in industrial processes or responsible for biogeochemical cycles. Microbial metabolism in extreme environments has two defining characteristics. It is slow and it trends towards the lower measurement thresholds of all current techniques and methodological trends. This provides the crucible of challenge to drive novel techniques, forcing researchers to employ new ideas, as well as reinvent old ideas that have fallen out of favor or become overshadowed by current buzzwords in the field. Some of the techniques that have been creatively harnessed to successfully shed light on the impact of microbial metabolism in atypical environments include microscopy at surface, air interfaces, mass balances, radio-labeled fatty acid synthesis, and stereoisomeric ratios. The divergence between the rate and career span of the researcher and the rate of the subject matter is a central consideration in this context and can be overcome by intentional and rigorous long-term experimental design, mathematical modeling, teamwork, global perspectives regarding measurement and impact. All microbial metabolisms can be arranged according to three principles. First, how the organisms contain carbon for synthesizing cell mass. The organism can contain carbon in three different ways. First, autotrophic. Carbon is contained from carbon dioxide. Second, heterotrophic. Carbon is contained from organic compounds. Third, mixotrophic. Carbon is contained from both organic compounds and by fixing carbon dioxide. Second principle, how the organisms obtains reducing equivalents, hydrogen atoms or electrons, used either in energy conservation or in biosynthetic reaction. First, lithotrophic. Reducing equivalents are obtained from inorganic compounds. Second, organotrophic. Reducing equivalents are obtained from organic compounds. Third principle, how the organisms obtain energy from living and growing. First, we have phototropic. Its energy is contained from light. Second, we have chemotropic, which energy is contained from external chemical compounds. In practice, these terms are almost freely combined. Typically, examples are as follows. First, we have chemolithoautotropes, obtain energy from the oxidation of inorganic compounds and carbon from the fixation of carbon dioxide. Example, nitrifying bacteria, sulfur oxidizing bacteria, iron oxidizing bacteria, and null gas bacteria. Photolithoautotropes obtain energy from light and carbon from the fixation of carbon dioxide using reducing equivalents from organic compounds. Chlorobaiaceae, chromothiaceae, hydrogen sulfide or H2S as hydrogen donor, chloroflexus hydrogen 2 as reducing equivalent donor. Chemolethoheterotrophs obtain energy from the oxidation of inorganic compounds but cannot fix carbon dioxide. Examples, some Theobacillus, some Begaia toa, some Nitrobacter spp, Wollinelia, some null gas bacteria, some sulfate-reducing bacteria. Chemoorganoheterotrophs obtain energy, carbon, and hydrogen from biosynthetic reactions from organic compounds. Examples: most bacteria like Escherichia coli, Bacillus, SBP, and Actinobacteria. Some microbes are heterotrophic, more precisely chemoorganoheterotrophic, using organic compounds as both carbon and energy sources. Heterotrophic microbes live off of nutrients that they scavenge from living hosts or find in dead organic matter of all kind. Microbial metabolism is the main contribution for the bodily decay of all organisms after death. Many eukaryotic microorganisms are heterotrophic by predation or parasitism. Properties also found in some bacteria such as Diluvibrio, an intracellular parasite of other bacteria causing death of its victims, and myxobacteria such as Myxococcus, predators of other bacteria, which are killed and lysed by cooperating swarms of many single cells of myxobacteria. Most pathogenic bacteria can be viewed as heterotrophic parasites of humans or the other eukaryotic species they affect. Heterotrophic microbes are extremely abundant in nature and are responsible for the breakdown of large economic polymers such as cellulose, chitin, or lignin, which are generally indigestible to larger animals. Generally, the oxidative breakdown of large polymers to carbon dioxide requires several different organisms, with one breaking down the polymer into its constituent monomers, one able to use the monomers and excreting simpler waste compounds as byproducts, and one able to use the excreted wastes. There are many variations of this theme, as different organisms are able to degrade different polymers and secrete different waste products. Some organisms are even able to degrade more recalcitrant compounds such as petroleum compounds or pesticides, making them useful in bioremediation. Photoorganoheterotrophs obtain energy from light, carbon, and using equivalents for biosynthetic reactions from organic compounds. Some species are strictly heterotrophic. Many others can also fix carbon dioxide and are mixotropic. Examples: Rhodobacteria, Rhodopseudomonas, Rhodospirillum, Rhodomicrobium, Rhodocyclus, Heliobacterium chloroflexus. Alternatively, the photolithoautotropy with hydrogen. The process of making and breaking down carbohydrate molecules illustrate two types of metabolic pathways. A metabolic pathway is a step-by-step series of interconnected biochemical reactions that convert a substrate molecule or molecules through a series of metabolic intermediates, eventually yielding a final product or products. For example, one metabolic pathway for carbohydrates breaks large molecules down into glucose. Another metabolic pathway might build glucose into large carbohydrate. The first of these processes requires energy and is referred to as anabolic. The second process produces energy and is referred to as catabolic. Consequently, metabolism is composed of these two opposite pathways. First, anabolism, building molecules. Second, catabolism, breaking down molecules. Anabolic pathways. Anabolic pathways require an input of energy to synthesize complex molecules from simpler ones. One example of an anabolic pathway is the synthesis of sugar from CO2. Other examples include the synthesis of large proteins from amino acid building blocks and the synthesis of new DNA strands from nucleic acid building blocks. These processes are critical to the life of the cell, takes place constantly, and demand energy provided by ATP and other high-energy molecules like NADH and NADPH. Catabolic pathways. Catabolic pathways involve the degradation of complex molecules into simpler ones. releasing the chemical energy stored in the bands of those molecules. Some catabolic pathways can capture that energy to produce ATP. The molecule used the power of cellular processes. Other energy-storing molecules, such as lipids, are also broken down through catabolic reaction to release energy and make ATP.